Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, I'm going to talk about dideoxy sequencing, which is also known as Sanger sequencing. This is one of the basic and fundamental idea behind a DNA sequencing process provided by Fred Sanger. Okay, so let's talk about dideoxy DNA sequencing. Now, DNA sequencing means you need to know exactly what are the nucleotides and their position. That's what we know as a sequence, right? Because if I only tell you that a DNA is prepared with A, C, C, T, A, G, right? Or let's say if I say you that the DNA contains 2A, 3Gs, 5C, it's not going to give you any data about the sequence. To tell you about a DNA sequencing, you should always start with a specific end to a specific end, and also tell you the position of each nucleotide. So now, if I tell you this, that 5 prime A C C G T A C 3 prime. This is an information that is going to tell you, yes, this is the exact sequence of the DNA. But if I tell you there is only 3C, 1G, 2A, 1T, it's not going to give you any information. So how exactly you will know what is the sequence of a DNA, which is very, very important for anything that we study to understand the presence of DNA and genes that we know this day, based on the understanding that we know the sequence of the DNA. And dideoxy sequencing or Sanger DNA sequencing is one of the methods that will give you a unique sequence of a DNA. Now, how the process is actually done? The process is using a specific modified version of nucleotide sequences known as dideoxynucleotides. Normally, the nucleotides present in the DNA is known as deoxynucleotides. Because deoxynucleotide means it's a modified version from a ribonucleotide which is present in RNA. Deoxyribonucleotide is simply it does not carry oxygen in the 2' of the ribose sugar. While we talk about dideoxy, di means 2, deoxy. So actually this type of ribonucleotide, this type of nucleotide sequence, what you'll find is 2 oxygen missing. One is from 2', that's for a normal DNA, but another oxygen missing from the 3' hydroxyl, like 3' of the ribose sugar as well. If you look at the ribose sugar, let me draw the ribose sugar, something like this. This is 1, this is 2, 3, 4, 5. So there are these 5 separate regions of the carbon. Now this 1, 2, 3. So this 2 normally for RNA contains hydroxyl, 3 also carries hydroxyl. For DNA, 2 carry only hydrogen, no oxygen. That's why we call deoxyribonucleotide. But in case of dideoxy, this oxygen is also missing from the 3'. And if you remember how the backbone of DNA is actually formed is with this 3' and 5' of the corresponding, the next ribose sugar. So the phosphodiester backbone is actually formed by linking the bases with one of the 3' and another one is the 5'. So this is how the interaction actually take place, right? 5' and 3' of the ribose sugar is really, really important to make the chain of nucleotides to form actual DNA sequence or one single strand of the DNA. But if there is no oxygen present in the 3' of the ribose sugar, then that ribose sugar will never elongate further. So whenever during the synthesis of DNA, we incorporate dideoxynucleotide, it will terminate the replication because there is no room for adding another ribose sugar, another nucleotide to it. That is the beauty of this sequencing method. Frederick Sanger exploited this idea of utilizing dideoxynucleotides to intentionally terminate growing strand of the DNA to find out the sequence of the DNA. How exactly? Let's look at it. Let's say in this case we want to find out an unknown DNA. We don't know the sequence of the DNA. But for our understanding, I put this is the sequence, let's say, is there, but we don't know it. So how can we know? Now let's say what we do here is simply we take that unknown copy of the DNA and we put that unknown copy of the DNA in different reaction vessels. And actually there are 4 different reaction vessels that are used. by Fred Sanger. Let me draw them on the same length, separate. Okay, 4 separate vessels he used, and actually what he did is in all the separate vessels he put all the normal version of nucleotides like ATP, GTP, TTP, CTP, all those things. Those are all present in all this tube. We have GTP, ATP, CTP, TTP, and all of them are DGTP, DATP, DCTP, DTP. Okay, for the DNA nucleotides, they are already present. The unique feature is she added dideoxynucleotide sequences one at a time for each of this specific reaction sample. For example, in this first one, he put DATP. Second one, DGTP. Then CTP, TTP. DD means dideoxy. So in this all these tubes, they carry 4 normal version, but apart from one normal version is removed. That means in this first tube, they have normally DGTP, DTTP, DCTP, but all DATP. Is it clear? 3 normal, 1 dideoxy. Similarly, for this next one, if I remove this, for this next one, all of them will be normal, like the ATP, CTP, TTP. Those are normal. GTP is separate. This one, ATP normal, TTP normal, GTP normal, CTP is dideoxy. This one, again ATP normal, CTP normal, GTP normal, TTP is dideoxy. So for each of this tube, one of the nucleotide is dideoxy in nature. So what we will do here, the reason behind this, in all this tube, he put the unknown DNA fragment. So let's say this is the unknown DNA fragment in all of them. Okay. In this unknown DNA fragment, let's say there are length of other segments are out there, but we only want to know the sequence of this region. So what we consider is that we also need to add some primers. So primer is added, no issue with that. Primer added. In all this case, primer added. And then it comes down to this part, that is A, C, C, G, T, A, C. This is the sequence. We don't know that though. Then what we do is simply, we know that all the normal version are present except for AT. So what will happen here? At the very beginning, the complementary of the sequence we want to find out. So complementary of A is T, normal T is present, so T will be added. Complementary of C is G, so that is normally present, added. Again, G normally added. Complementary of G will be C, which is also present normally, added. Complementary of T will be A. But in this case, no normal ATP is present. Dideoxy ATP is present. So now, the dideoxy ATP, so dideoxy A will be added. And what we know, as it is dideoxy in nature, it will terminate the polymerization of DNA at this point. So no further DNA will be produced. So what we'll see here? In this case, the length will be terminated here. It will not produce the rest of the DNA fragment. The same thing happens for each of this tube in presence of DGTP, DCTP, DTTP. For all this tube, the same reaction runs like that. So if you run reactions like that, then what will happen? Try to understand, for each of these fragments, what we will generate. Let me show you with a simple drawing here. Let's assume, in this case, if we use DATP, then the fragment length that we produce will be shorter because this is the point where it will be terminated. So length will be 1, 2, 3, 4, 5. Okay. So we have this length of 5 with dideoxy ATP. Then the second reaction, if we use with DDGTP, then what will happen? DDGTP means opposite and complementary to C. So you'll get one this band, one this band. So one band with 2, another band with 3. Okay. Then if we use DDCTP, only one G is present, so it will terminate here. So band for 4. Okay. And then also, sorry, for GTP, we have this 2 as well as another one because C is also present there. So 1, 2, 3, 4, 5, 6, 7. So 7 is also there. And for CTP, it's only 1. And for TTP, we have fragment 1 and 6. So these are the different fragments that we'll produce. If you don't understand how I draw all these fragments, try to understand here. I'm going to explain it again. Because no, every single time there is the complementary dideoxynucleotide is added, the chain is terminated. That's why you also call it chain termination method of DNA sequencing. So if we use DDTTP, the chain will be terminated right here. So if you use TTP, the length will be the smallest because of one nucleotide will be produced only. If we use DDGTP, then it will terminate after this C, so length will be 2. Again after 3, so length will be 3, and again after the end, that is after 7, so length will be 7. All if we use DDGTP. If you use DDATP, then complementary of ST, so it will terminate here, so it will be length of 5 nucleotides. So that's how we get different length fragments from different reaction mix from different tubes. So all the fragments from the tube are generally taken here, and actually for each and every separate tube we load them in a agarose gel in different groove. So what we mean by the groove, let me show you here. We take the agarose gel, and in this agarose gel, what we put? We simply put the 4 separate grooves for A, G, C and T. A, G, C and T. And let's say, what we will see now, for DDATP, the band that we got here is for 5. So let's say here, in these agarose, what we know is that the smallest band will migrate the further, and the tallest and largest one is the less. So here let's say 0, 1, 2, 3, 4, 5, 6, 7. These are the different length of the band that we are considering. So what are the bands that we'll observe? For taking DDATP, the band is only one band for the 5. So for 5, only one band. Okay. Now we go for what? DDGTP, 3 bands, 1 in 2, 1 in 3, 1 in 7. So 1 in 2, 1 in 3, 1 in 7. Then DDCTP, only one, that is in 4. So 4 DDCTP, and DDTTP 2, one is in 1, another one is in 6. So this is the band pattern that we observe. This is the band pattern that we observe. Once we load all this reaction mix into the grooves and we run the gel, we got the band. Now it's time to find out where exactly, what is exactly the sequence of the DNA. Because here to understand the situation we know the sequence, but actually we don't know. So what we want to find out is the sequence. Now how to read the sequence? We read the sequence from the smallest one to the largest one. And smallest one to the largest one will be 5' to 3'. That's what we are going to find out now. Okay. So let's find it out from smallest to the largest. So let me erase this part. So what we are learning from. So now once we see all the band patterns, now what we need to find out the exact sequence of the unknown DNA. Because although in this case we draw the unknown DNA, actually this system should be different. It should never be 5'3". It should be 3', 5' actually. Because we are trying to find the sequence in this direction always. And the DNA replication in the new strand will continue from 5 to 3. So actually the sequence should be 3', 5' there. So if you want to find this sequence, that is unknown to us. We don't know that sequence. So how exactly we'll know? We run the process with DDNTPs in 4 separate reaction mix. We load it into the agarose gel. We run the gel. We find a band pattern. Now from this band pattern, we want to find out the exact sequence. Now remember always the band pattern that we get. The shortest band corresponds towards 5 prime and the tallest band corresponds towards the 3 prime. So you'll always go like 5 prime. This is 3 prime. Okay. So now let's look at here how exactly we take the band and finding out the exact unknown sequence. Try to remember one thing. In this case, whatever sequence we got, we take that sequence as a template and we add DNTPs and DNTPs only. 3 DNTPs and 1 modified DNTPs in each reaction mix. So every time there is a DNTP or didioxy DNTP is added, it terminates the DNA, right? That's how we'll get different fragments which we'll see in this band. So whatever sequence we'll get directly from this band pattern is not the exact DNA sequence. It's a complementary of that DNA sequence, right? So let's look at it in the first place. So what we'll see here is simply we start with the shortest going towards the largest. So shortest one is 1 and that is T. So you go up with T. This one. Then we go with 2 which is G. Then again 3 which is also G. And then we go to the next which is C. Then next A. Then next T. And then next G. And we know the sequence ordered from 5 towards 3. So this is a complementary sequence of the actual unknown DNA. So once we find the complementary sequence, it's now very easy to find the real sequence. Simply make a complementary of that copy. A, 3 prime will be there, which is opposite complementary to the 5 prime. So A, C, C, G, T, A, C. If you look at here now, A, C, C, G, T, A, C, A, C, C, G, T, A, C. That's how we exactly find out the unknown DNA. Okay. So whatever unknown DNA you take, you use the Sanger method, which is very simple but very effective way to find out exact unique sequence of the unknown DNA copy by simply using dideoxynucleotides. So what we've done, we simply took the unknown DNA fragment, we add some primer, we add DNTPs in each separate reaction mix. We have 4 separate reaction mix, and in each of this reaction mix we have 3 normal version of DNTPs and 1 dideoxynucleotide. Because right after the dideoxynucleotide there is a chain termination which we can easily get by loading into the gel. We get the band pattern. From that pattern we get the complementary copy of the unknown DNA. From there we easily find out the unknown DNA sequence. So this technique is best when we apply it for short stretch of nucleotide sequences. That's why Sanger sequencing is very good for any short segment of DNA sequencing. But if we talk about longer, taller segment of DNA sequences, this method is not so good. Because for the long sequences, it raises so many complications because this is time consuming. For every single round for a stretch of nucleotides, you need to run it several times of reaction mix, so many DNA, so many DNTPs, DDNTPs, cross reactions are possible. So the taller, the longer length of the DNA you take for the Sanger method, the more difficult it gets. So that is the only big disadvantage of DNA sequencing with Sanger method or dideoxy method. Apart from that, dideoxy sequencing method is one of the very best type of unique sequencing method that we have. So while we knew this method earlier, but while try to understand the DNA sequence of human being during the Human Genome Project, it's very difficult to utilize Sanger sequencing because human genome is huge and utilizing Sanger sequencing, it will take years. So to prevent that, we utilize another method of DNA sequencing, slight modification of the Sanger method, which also involves Sanger method as a unique source of DNA sequencing. It's known as shotgun DNA sequencing. In shotgun DNA sequencing, we modify the Sanger sequencing a little bit, and then it allows us to finally sequence so many long length of the DNA, large DNA molecules. So in the next video, I'm going to talk about the shotgun sequencing of the DNA. So if you like this video of Sanger sequencing, please hit the like button, share this video with your friends, and subscribe to my channel to make and see more and more videos like that.